I found out in 1989 about a wonderful protein that was made in jellyfish. And this protein was a very interesting protein because it was fluorescent. That means that when you add light to it, blue light, you get green light out. And I realized this would be a wonderful way of marking cells and animals. And I actually showed that that was possible. And since then, over 100,000 people have used this, or 100,000 papers have been produced to watch various aspects of biology. We use this marker in many ways in the laboratory. We use it to study where genes are active. We use it to ask where proteins are made in cells. We also use it to label cells. And once you can see something, that means you can then look for variants, mutants that are different. And these allow us to study aspects of, for example, how nerve cells grow. So if we find a mutant in which the cell that we can see does not grow this way but grows this way, then we know we have a gene that is important for the growth of the cell. If we see that we have an animal that has more cells or fewer cells, we can then look at the gene, and that's a way of being able to ask the question, how is it that an organism can make different types of cells and the correct number of cells? My experience in the United States is that without the support of the National Institutes of Health, National Science Foundation, and private foundations supporting research, most of the basic essential research that has led to so many discoveries would not have been made. These days there's a very big push for research that goes directly to answering a particular question. I think this is misguided. I think people should be supporting that, but also and mainly supporting basic research. Well, it changed in many ways. First, I'm asked to do interviews like this. I've been asked to give talks at many places where normally I wouldn't have been invited before the Nobel. I've also had a chance to talk with many more students and try to get them excited about science. Those are some of the things that have changed. I do a lot more traveling. I've really enjoyed my life since the Nobel. It's a wonderful honor. I'm very grateful for it. And it's given me a chance to meet people that I normally wouldn't have done before. I've always had a mixture of research and teaching in my life. I've been at Columbia University for almost 30 years, and that has not really changed since getting the Nobel Prize. I still do my teaching, I still enjoy my teaching. I have students that also work in my laboratory, and I'm also going off and giving talks and doing the research or directing the research in the laboratory. So it's always a mix of these things, and it hasn't really changed.